So hey learners, welcome and welcome back to the part 3. In this lecture, I'm going to make sure that I will finish all the remaining topics here. So without wasting any further extra time, let's get into the chapter. So the topics to be covered in this chapter are enzymes, primary and secondary metabolites, dynamic state of body constituents, which is the concept of metabolism, metabolic basis for living, and the living state. So let's start with the enzymes. Now, what are enzymes? Enzymes are the biocatalyst made up of proteins. Now, you may know what are catalysts. Catalysts are actually help to increase the rate of any chemical reaction. Then, what is biocatalyst? It also helps to speed up the reaction in any living system. Okay, they are made up of protein. They are usually proteins, and they also act as catalyst because they help in the reactions happen more efficiently without being used up and changed in the process of any reaction. All enzymes are proteins, but all proteins are not enzymes. This statement is very important to understand the concepts of biochemistry. See, most enzymes are made up of proteins, and we know this. But there is some RNA molecules which can also act as enzymes. These are called the ribozymes. And here comes our exception. These RNA based enzymes are known as ribozymes. It is present in ribosomes, and you will also see in the chapter molecular basis of inheritance the topic translation 23 rRNA. And other than that, it also helps in the process of splicing where it acts as a RNA catalyst. Enzymes are water soluble and they only function in the aqueous medium like blood or cytoplasm inside the cell organelles, and the reasons are very basic. They are made up of proteins with their hydrophilic amino acids on their surface. And these amino acids form the hydrogen bonds with water, making the enzymes dissolve easily. And why solubility matters to the enzyme? Because enzyme needs to move freely to find the substrate easily, and while being water soluble they ensures that the interaction with the substrates become easy, and it also helps to maintain the proper shape and activity of the enzyme while being soluble at the same time. Well, there is also an example from the chapter sexual reproduction and flowering plant where we decrease the content of water by 10 to 15%, while to the growing seeds they need a less content of water. For the proper development of seeds, it is needed to decrease the activity of the enzyme. Now we have a topic to be covered, which is biological importance of enzyme, where we have few examples like digestion, where the breakdown of macromolecules take place. For the example like amylase, or you can say salivary amylase, which is present in the saliva, which break down the starch into the glucose molecules in our mouth. This is the most common example. Lipase to break down fats into fatty acids, and proteases for the breakdown of proteins into amino acids in the digestion. Then we have DNA replication and repair, where DNA polymerase or ligases helps in the copying DNA during the cell division. Then the metabolism, where the enzymes regulate metabolic pathways, for the example glycolysis Krebs cycle. Then we have catalysis of biochemical reactions, where enzyme accelerate the rate of chemical reaction by lowering the activation energy. I mean, without enzyme you can't even imagine to sustain life, because most of the reactions in the body would be too slow. Enzyme also help to detoxify the harmful substances from our body. Now, the catalysts are generally classified into two main types, into organic and inorganic catalyst. Now, here is a breakdown of each one of them. So, organic catalyst also called as enzymes, and they are primarily made up of carbon based compounds, and they are often used in organic chemistry and in biological system. For the examples, you can use any of these proteins like pepsin, trypsin, and exceptions like ribozymes. Then we have inorganic catalyst. Inorganic catalysts are composed of metals, ions, or any molecules or metal containing compounds. They are widely used in industries or environmental purpose. For the examples like nickel, silver, and etc. And even they can be prepared in the laboratories also by our scientists. Enzyme catalysts are different from inorganic catalysts in many ways, but one major difference that your NCERT also mentions is that the organic catalysts like enzyme work under mild or biological conditions. For the example, like any of the thermophilic organism, suppose in human body the temperature could not be above 40 degrees Celsius, because after that it could be damaged. What I mean to say is most of the enzymes are proteins, and protein can be easily denatured or can lose their functionality after a certain level of temperature and pH. But in an organic catalyst, they can function under a wide range of conditions, including extreme temperature and pH. Because thermal stability is thus an important quality of such enzymes. For example, like Thermus aquaticus bacteria, which has Taq polymerase enzyme in it. This bacteria with these special abilities was first discovered at California's Yellowstone National Park in their volcanic eruption. And you will also gonna read the same thing in PCR Biotech chapter. And another example is Archaebacteria. They also have some kind of features like this. However, other than these organisms, we have few other examples who lives under extremely high temperature, such as hot winds under the deep ocean. It's a actually crack or opening kind of thing in the sea floor. And another is sulfur springs. It's a natural water source rich in sulfur, as the name suggests. And important thing is they are stable in the high temperature and retain their catalytic power even at the high temperature like 80 to 90 degrees Celsius, so that they can work at any temperature. And the third point is enzymes are highly specific in nature for just one reaction. I mean, they often act on only one type of substrate. But for the inorganic catalyst, they are less specific in nature. They can speed up many different reactions, and they are generally can facilitate various reactions at a time. The fourth point is enzymes could be regulated by inhibitors and activators etc. But for the inorganic catalyst, they are not usually regulated in the same dynamic way as the enzyme does. The fifth point is that the enzymes required water for their functionalities, while the inorganic catalyst doesn't need any water to function. And one thing which is common in organic and inorganic catalyst, that they are catalysts, so they're going to increase the rate of reaction in both of them. And second of all, they are going to be unchanged at the end of the reaction, because the only thing they're going to do is to increase the rate of reaction, not going to used in the reaction. Now, before understanding enzymes, let's first see what are chemical reactions. So we already know what are these from the very previous classes and basics. It's so basic, but let's see an overview of it. So when chemical compounds undergoes two type of changes, the first is physical change. When a physical change simply refers to a change in shape without breaking the bonds in it. It's a complete physical process where change usually they are reversible and new products are not formed, and the substances retain its chemical properties. And we have another change is chemical change. Okay, when bonds are broken and new bonds are formed during the transformation, this will be called as chemical changes. Okay, when change in chemical structures and compositions of matter, they are actually what chemical change. Okay, they are usually irreversible, and one or more new substances are formed. And whenever the new substances are formed, they have the different properties from the original substances. Few examples of them are given in your NCERT, like when ice melts into water and when water becomes water vapor are the examples of physical change. And the example of inorganic chemical change is when barium hydroxide reacts with sulfuric acid for the formation of barium sulfate and water. This is already a balanced chemical equation. Hydrolyzation of starch into glucose is an example of organic chemical reaction where starch is a polysaccharide broken down into the different monosaccharide units, mainly glucose, in the presence of water molecule. These both examples of chemical reactions has given in your NCERT just to specify the rate of a physical or chemical process refers to the amount of product formed per unit time, which is also called as speed of reaction. Let's see on the another slide. It can be expressed as the speed of reaction or the rate of reaction, which is product formed upon time. So what enzyme actually do here? Enzyme increase the rate of this reaction without getting consumed in it. That is what I'm trying to make you understand from starting of the lecture. The rate of reaction is not just for chemical changes but also for the physical and chemical both the changes. It So the enzymes act upon both accordingly. It changes the state, shape of the physical changes, but it changes the nature chemically in chemical change. Lock and key hypothesis. It's a model was introduced in early 90s. Actually, this model explains how enzyme and substrates interact during a biochemical reactions. As you can see on the image, this is our enzyme which is analogous to lock, and this is our substrate which is analogous to key. Now, according to this hypothesis, the enzyme's active site has a specific shape that exactly matches the shape of its substrate, just like the key fits perfectly into a lock. Only the correctly shaped substrates can fit into the enzyme's active site to form the enzyme substrate complex. But later on, and that was the major drawback of this model. This model says that enzyme's active site is rigid in nature and doesn't change its shape. And but in reality, enzymes are too flexible. In fact, many enzymes can adjust their shape, can change their shape slightly just to fit the substrate in it. This is ignored by this model. This model found to be less accurate to explain all the enzymes. Then we came in notice to the another fit and induced fit hypothesis, which says that when a substrate approaches an enzyme, the enzyme's active site is not a perfect fit at first. Okay, it's not a perfect fit at first. Instead, the enzyme undergoes a conformational change, a slight change in shape to better accommodation the substrate. This induced fit allows for tightly binding and a more efficient catalysis of the reaction. Even this hypothesis is more accurate than the previous one and widely accepted in all over the world today. Now let's begin by the structure of an enzyme. On the screen you can see this is a typical 3D structure of an enzyme. I have used this image so that you will get a clear classification of the enzymes. Now the enzymes are typically globular proteins with a complex three-dimensional structure. When I say globular protein, then you must remember those proteins which you read in the last video about the amino acids and their 3D structures, and that 3D structure is actually what enzyme has. It is related to it directly. Now you can easily determine by the shape and structure of an enzyme that how and when they interact with the specific substrates. Structure of an enzyme can be described at 4 different levels. We will see each one of them one by one. The first one is primary structure. If you remember the sequence of an amino acid in the polypeptide chain, their importance, they determine how the enzyme will fold upon itself and as a functional shape. After that, there is a secondary 2-degree structures of protein where alpha helix and beta plated sheaths were stabilized by the hydrogen bonds between them just to provide the initial folding that leads towards the overall 3D shape, but it is not the actual 3D shape, it is just the foldings. Then there is a tertiary structure which is 3D in shape, determined by many bonds between them, which is an actual shape of enzyme. And if you remember, majority of proteins and enzymes in the protoplasm exhibit these tertiary structures only. Any protein with the primary and secondary structures, they are not the enzymes, but when you look at the tertiary structures, you will notice that the backbone of the protein chains actually folds upon itself. The chain crisscross in their itself by their own, and hence many crevices and pockets are made. One such pocket will be called as active site. We will see what active site actually is. So, when an active site of an enzyme is a pocket or crevices into which the substrate actually fits, and the reactions occur at a very high rate. So, an active site has a very unique shape where the substrate fits perfectly into it, and it may change its shape slightly just to fit the substrate perfectly in it. You already know because of the induced fit hypothesis. And as soon as substrate binds to the enzyme's active site, enzyme starts catalyzing the reactions at very high rate. As you can see on this image of enzyme, there are many pockets or crevices, or you may say cleft like structures on this enzyme, but there is only one crevices will behave like the active site, and that active site, that will be, there will be some particular R group. The R group in the active site of an enzyme refers to the side chain of an amino acid. These R group play many crucial roles, such as every shape of active site in an enzyme is different from each other because the shape of an active site is depend on the final folding of the protein. If anything disturbing happens with its folding, then the active site completely lose its function and the enzyme will be denatured as soon as the R group removed from that particular active site. We have many crevices formed on the active site, but there is only one crevice gonna be the active site. Now let's begin by the most important term, which is the mechanism of the action of enzyme. So I'm going to explain this whole mechanism, the catalytic cycle of an enzyme, and their actions can be described in the following steps, 4 steps. It will be covered in the 4 steps. So first is substrate binding, then the formation of transition state, then the catalyst and the conversion of substrate into the product, then the product will be released out. We will discuss all these 4 steps in detail, where these terms gonna be used, where E stands for enzyme, S will be substrate, ES is enzyme substrate complex, EP is enzyme product complex, then P for product. Okay, let's begin. Now, how do enzymes bring about such high rate of chemical convergence in it? Have you ever wondered? To understand this phenomena, we have to study enzymes a little more. We have already understood the idea of an active site or the chemical or metabolic convergence of a reaction. So for now, we will see the diffusion of substrate is directly proportional to the binding. It means when the rate of diffusion of substrate increases, then the chances of binding will automatically increase. There are some certain factors on which they will increase, but we will discuss these factors later on. When substrate binds to the enzyme's active site, first loose binding will occur. Then these binding of substrates induces the active site to alter its shape so that the final tight binding will occur. that leads to the formation of enzyme substrate complex. That is a reversible reaction because the enzyme and substrates bind together through the covalent interactions such as hydrogen bonds, ionic bonds, and many van der Waals forces. But these interactions are relatively weaker and they can be easily disrupted. So as you can see in the image, either the ES complex will remove back to the its original positions like enzyme or into substrate particularly, or it can be proceed up to the next step. So the next step will be in between the conversion of this ESEP complex, there is one more state forms. Actually, where at that position substrate is completely bounded to the enzyme's active site, where a new structure of the substrate called the transition state structure is formed. I was not trying to mention it here because that will create a confusion, but I am soon going to cover this in the next graph. Now the active site of the enzyme now in the close proximity of the substrate to change the substrate chemically. See how this will happen. See, first there is some R group on the active site which is going to break the chemical bonds of the substrate. After all this bond breaking and making process, there will be some physical change in the substrate that will lead to the formation of a new enzyme product complex, and soon after that the enzyme releases the product of the reaction out and the free enzyme is released to bind the another molecule of the substrate and run through the catalytic cycle once again and again after repeatedly. In short, symbolically this whole procedure can be depicted as the conversion of substrate into the product. Now I am going to summarize everything through this graph. So this graph also becomes very important to the point of view of your NCERT. Now see, the graph of activation energy typically shows the energy change during a chemical reaction. That is what we are going to discuss in it. And that is the reason why it also called as reaction coordinate diagram or energy profile diagram. Now as you can see on the x-axis we have reaction process or reaction coordinate, and which represents the pathway from reactant to the product formation. This substrate is compared to the reactant in any chemical reaction. Now on the y-axis we have potential energy in the unit kilojoule per mole. And one more thing, as you can see on this graph that the substrate always starts from certain energy level but not from the zero. You can find the reason in a cell's cytoplasm the molecules are always in some random Brownian movement because they always have some certain amount of energy in them. That is the reason why the substrate doesn't start from the zero. Now we will see the pathway of how substrate actually converts into the product. So we have substrate on the starting energy level and product on the final energy level after the reaction occurs. So as you can see on the y-axis, the substrate, the energy of substrate is higher than the energy of product. So it's an exothermic reaction. By this you can also find the change in enthalpy, which is delta H, by the difference between the energy of substrate and product. When delta H is less than 0 in the exothermic reaction, you don't need to supply any energy in the form of heat in order to form the product. And the curve always goes up, then it drops down below. But it is vice versa in the case of endothermic reaction graph. Now the substrate cannot directly transformed into the product. It has to travel some energy barrier which is called the activation energy. Activation energy is the average energy difference between the substrate and the transition state. Even your NCERT says that the average energy constant is the difference of average potential energy of substrate minus transition state energy. Now this is one of the function of enzyme, that to convert substrate into product easily, enzyme always degrade the value of activation energy. To travel through this activation energy barrier, substrate must go through this transition state, intermediate transition state, which is the highest energy point among the whole reaction pathway, which stays just for the fraction of a second, and where the molecules are really in a high metabolic flux also. Other than that, there could be many more intermediate structural states or altered structural states which are very unstable in their structure between the stable substrate and the product. Stability is something which is directly related to the energy status of the molecule and their structure. And here in it, it is unstable because of this high energy content in it, as I already said. Now we will see what enzyme actually does here, being as a catalyst in this reaction, and how they are actually increasing the rate of the reaction. Enzyme actually gives an alternate path to this reaction by decreasing the energy of this transition state, so that the substrate can easily convert into the product without climbing this huge activation energy mountain or graph, you can say. Now we have two different type of potential energy diagrams given in your NCERT. One is exothermic and another is endothermic. So in the exothermic or spontaneous reaction graph, products have lower energy as compared to the reactant, as you can see in the image, and energy is always released in the form of heat, light or anything, etc. But the curve goes up to the activation energy and then it suddenly drops down. Now we have endothermic or energy requiring reaction, in which product always have the higher energy as compared to the reactant, or you can say substrate, but the energy is always absorbed in this reaction. Either it may be any energy, but it will be absorbed, and the curve goes up to the activation energy and stays at the higher energy state or the transition state. Then we have to discuss about 5 features of the enzyme. As per your NCERT says, we have chemical nature of enzyme and medium in which it acts, specificity and selectivity, and then we have turnover number at the end. We'll discuss each one of them with examples, features in detail. So let's begin. So let's begin with the specificity of enzyme. Actually, enzyme's active site is highly specific to one substrate or a group of similar substrates, you can say. Actually, what happens here is enzyme's active site has some fixed type of chemical groups present on them, which leads to some fixed type of chemical reactions to be occurred on the enzyme's active site. But sometimes we have some specific inhibitors with the same shape at the same time as the active site has, but they are chemically different, so they can't react with each other and can't form the further products. Just like a key fits in a specific lock, similarly enzyme can act on only specific substrates. Enzyme selectivity refers to the enzyme's ability to choose a specific substrate or reactant out of many possibilities when multiple similar options are available. So we have the same analogy here. A key that opens only one lock has specificity of enzyme, when a key that fits into many locks but opens one better than others shows the selectivity of nature of the enzyme. This is an example of metabolic pathway when one molecule of glucose is broken down into two pyruvic acids by the process called glycolysis, and this reaction is catalyzed by a series of 10 different enzymes within this process. And this reaction is also from the respiration chapter 14, you will study there. By the use of one substrate, we can form three different possible products. For example, under normal aerobic condition, by the use of pyruvate dehydrogenase enzyme, we will get acetyl-CoA. And for the another example, in our skeletal muscles, under anaerobic condition, by the use of lactase dehydrogenase enzyme, we'll get lactic acid. And similarly, during fermentation or by the use of pyruvate dehydrogenase enzyme, we will get acetaldehyde, which is also known as ethanol. It's an alcohol. So as you can see in this reaction, when glucose turns into pyruvate, by the use of one substrate, we can form three different possible products. But as soon as we add enzyme to the reaction, in different condition, different products are possible. So the conclusion is, out of various possibilities, enzyme can select any one out of these three products. Unlike organic chemistry, there is no major minor products going to be formed. But then we have metabolic pathway, a multi-step series of chemical reactions when each of these steps is catalyzed by the same enzyme complex or different enzymes are called metabolic pathway. And there are 1,000 types of enzymes, each catalyzed a unique chemical reactions or metabolic reactions, you can say. And there is one condition, that if the enzyme is not selective and not fixed for one product, then this metabolic pathway reaction will not move forward. So in this mechanism, our body controls the metabolic pathway by using the end product of the pathway. Actually, the end products can slow down or stop the process when enough of it is made. This helps to maintain the balance and also to prevent the wastage. And we are also going to see this feedback mechanism technique in class 12th human reproduction chapter also. Now we will see what is the turnover number of an enzyme. So basically, the turnover number of an enzyme, also called kcat. Kcat stands for catalytic constant. So basically it tells how many molecules of substrates can be converted into product by an enzyme in per second of time. It actually shows that how fast and efficiently one enzyme molecule can work. So, when an enzyme catalyzed reactions are carefully observed, we will get to know that catalyzed reactions are proceeded at a very high rate as compared to the uncatalyzed one. For this, we have an example from your NCERT, which is when carbon dioxide bonds with water. By the help of carbonic anhydrase, we will get carbonic acid, which is H2CO3. So, in the absence of an enzyme, the rate of this reaction is very slow, which is about 200 molecules of H2CO3 is being formed in an hour. However, by using the enzyme present within the cytoplasm of a cell, which is called carbonic anhydrase, the reaction speed dramatically increases by the 6 lakhs of molecules being formed every second. I mean, can you see this? Without the enzyme, it takes 1 hour to form 200 of molecules. But only a single molecule of enzyme can able to form 6 lakhs of molecules in just 1 sec. I mean, you can see this is how incredible it is. The enzyme has accelerated the rate by at least 10 million times. I mean, this is how powerful an enzyme is. So start taking enzyme very seriously than your own existence. And yes, carbonic anhydrase has the highest turnover number among all the other known enzymes. And it is also vital for the process like breathing and pH regulation. So you always remember the name of this enzyme, carbonic anhydrase. And most of the people get confused here that the enzyme can make a reaction go faster and can speed up but can never start a reaction by its own. So always remember that one thing. So the formula to find the turnover number in second is Vmax upon E total. Vmax stands for maximum reaction rate and E total is total enzyme concentration. Although you don't have to do the questions related to this formula because it is not in your NCERT, but definitely you will deal with it in your further classes and 12th maybe. Now the enzyme's activity is directly influenced by several key factors that can either increase or decrease the rate of enzyme's catalytic reactions. which can alter the 3D structures of protein, because enzymes are indirectly proteins. So these include temperature, pH, change in substrate concentration, or you can say binding of specific chemicals that regulate its activity. So we will see. Let's begin by temperature because it plays huge role in it. Enzymes generally function in a narrow range of temperature, and there is a term called optimum temperature, which means like each enzyme shows its highest activity at a particular temperature, which is called the optimum temperature. Organisms like cold-blooded and warm-blooded, there are many of the organisms who has evolved over time as the 37 degree as their optimum temperature, in which their enzymes can work maximum, and even in human 37 degree is the optimum temperature at which almost all of the enzymes can work very efficiently. Now the activity of enzyme declines both below and above the optimum temperature value. In the low temperature, they preserve the enzyme in a temporarily less active state because of the increase in kinetic energy, where molecules move less and more frequent forceful collisions between enzyme and substrate. Whereas high temperature can destroy enzymes activity by slowing down molecular movement and reducing reaction rate. Hold on, let me explain this high temperature. As soon as the temperature rises above the optimum temperature, the activity rapidly decreases, such as the active site loses its shape and substrate is no longer able to fit into the active site, and enzyme also become non-functional and inactive. So let me clear it with few examples for all of these three temperatures. Pepsin for the optimum temperature, which works best at 37 degrees Celsius. Then for the higher temperature we have thermophilic bacterial organisms like Tech polymerase, if you remember, which works at 70 degrees Celsius temperature or higher. For the low temperature, I really can't sure about the examples because enzymes work really less actively at low temperature, but still you can check out your refrigerator, maybe you find some bacteria out there, I'm not sure. Okay, leave. Now we have this general thumb rule which says that the rate doubles or decreases by half for every 10 degrees Celsius change in either both the directions. The value of this Q10 is equal to 2, where Q refers to the temperature coefficient and 10 indicates the 10 degree temperature difference. We will see how it works. If the temperature increases by 10 degree, then the enzyme's reaction rate roughly doubles. Suppose at 20 degrees Celsius the reaction rate is 1 unit, and when it is 30 degrees Celsius the rate becomes 2 unit. It doubles, it increases because the molecules have forceful collision between the enzyme and substrate, and kinetic energy increases. And when the temperature decreases by 10 degree, the reaction rate halves. Again, you just have to reverse this value of Q10 is equal to 2. Suppose at 30 degrees Celsius, the rate becomes 2 unit, and decreasing to 20 degrees Celsius, the rate automatically becomes to the 1 unit. That is it for temperature. The rate of reaction is half here because of the molecules move slower and they need less energy for bond making and forming products, and also you will see few collisions occur between the enzyme and substrate. Now we have this enzyme activity versus temperature graph. It helps to explain how enzyme activity changes with the temperature. So as you can see on the screen, we have this bell shape curve. So on the left side of this curve, it has low temperature region, about 0 degree to 25 or 30 degrees Celsius. Now as soon as the slope rises to the peak, it reaches the optimum temperature, which is about 25 degrees Celsius to 37 degree. Now the falling slope of above optimum temperature, which is about 40 degree to 47 degree range. And you already know the effects of low and high temperature, when they increase and when they denature from the last slide, so I'm not going to repeat. Similar to temperature, enzymes also show their maximum activity at a particular pH called optimum pH. I can make you understand this thing by see, in the 3D structures of enzyme, the final folding of charges is balanced, and this balance is maintained at the optimum pH range where they functions the best. At the optimum pH, enzyme shape is maintained and they work maximum. For the example, we have three different enzymes which works best at their optimum pH. As you can see on this graph, we have pepsin, salivary amylase, pancreatic lipase. So on the x-axis we have pH ranging from 1 to 8, and on the y-axis we have rate of reaction ROR, which represents the enzyme activity. So it's a bell-shaped curve where the every enzyme has its own optimum pH. For now, let's talk about to the respect of pepsin enzyme only. So on the graph, the activity of enzyme declines if on the both of the below and above value of optimum pH, similar to temperature. So as the pH goes down or above the optimum temperature in both of the conditions, enzymes activity is low and they become denatured, lose its shape and functionality. Because at the low pH it becomes too acidic, and at the high pH it becomes too alkaline and basic conditions. As the enzymes are protein, so change in pH can alter the charge of amino acids, and especially those at the active site. Enzyme may stop working entirely if the pH is too extreme or too low. So it becomes important to maintain the optimum pH for the enzyme to function properly in a biological system. Let's begin by the substrate concentration. So the number of substrate is always greater than the number of enzyme. The number of substrate is almost double or triple as compared to the number of enzyme. The substrate move towards the enzyme by the process of diffusion, and if we increase the number or concentration of substrate, then the rate of diffusion will automatically increase, and as soon as the diffusion increases, that will lead to the more chances of enzyme substrate complex formation, and that will directly lead to the increase in velocity of the enzyme. Can you see this diagram given below? We have many substrates over only few enzymes are there. All over the substrates are in a competition to bind with the enzyme. Okay, let me simplify it through this graph. So on the x-axis of this graph we have substrate concentration, and on the y-axis we have velocity of reaction VOR, which represents the enzyme activity. This V versus S graph shows that how the enzyme activity changes as the increase in substrate concentration. With the increase in the substrate concentration, the velocity of enzymatic reactions rises at first. Then the reaction ultimately reaches the maximum velocity called Vmax, and this is because the enzyme molecules are fewer than the substrate molecules, and after saturation these molecules, there are no free enzymes molecules left to bind with the additional substrate molecules. So the reaction is not going to be exceed by any further rise in the concentration of the substrate. Now these are the points what NCERT says. I know you are still confused. Let me repeat it for you. Now look at this hyperbolic curve very carefully. See, at the initial phase when the slope rises at low substrate concentration, enzyme activity is increased sharply with a very high speed. But still few substrate molecules are still remain available because all the enzymes active sites are not occupied at this point. Now see, at the high substrate concentration, enzyme activity suddenly stops, with the speed also slows down because the all enzymes active sites are fully occupied at this point. They are saturated, and adding more substrate has no effect, no point on reaction rate. Also when the enzyme is working out its maximum rate called Vmax, and the graph suddenly becomes constant. But there is one interesting thing, that after doing all this we can't find a single substrate concentration at which Vmax is achieved. To understand this, let's suppose we have total enzyme is 100 where Vmax is achieved and all 100 are working. Then we will take half of this Vmax. The half of Vmax will be 50 enzymes where all the enzymes are functional. We couldn't be able to find the substrate concentration for 100 of enzymes. Then why not to find the substrate concentration for half of the Vmax? Something is better than nothing. So for doing that, try to point the Vmax graph to the substrate concentration. So finally, this is the substrate concentration where half of the Vmax is achieved, which is called as Km. And this Km will be 50 as the half Vmax is 50. Now you may ask, what is the significance of this Km? See, a better enzyme always have low Km because they provide high catalytic power. I will prove you why a better enzyme always have low Km as compared to high Km. Now as you can see on the screen, we have two different graphs with two different enzymes and the common substrate in both of them. In the first graph, enzyme is marked as E1 with a low Km, which shows a rapid increase in the reaction velocity, which indicates high substrate affinity to the enzyme, and enzyme binds to the substrate more tightly. And vice versa, in the second graph, enzyme is marked as E2 with the high Km, which shows a slower increase in the reaction velocity and also indicates low substrate affinity to the enzyme, where enzyme binds substrate loosely. So now can you answer me in which enzyme can form more products? Yes, you are right. Enzyme 1, because it has low substrate concentration, which gives more velocity to the enzyme and can form more product as compared to the second one. Okay, so now we move to inhibitors and how their presence can affect an enzyme. We'll see that. So the enzyme activity is also sensitive to the presence of some specific chemicals, or you may say inhibitors, which actually binds to the enzyme's active site and reduces or blocks its activity. Okay, so this complete process is called inhibition, and the chemical who done this inhibition called inhibitor. Now we have four different type of enzyme inhibitions along with their diagram. So we will see each one of them one by one. So first one is competitive inhibitors, then we have non-competitive inhibition, then uncompetitive inhibition or irreversible inhibition. Let's begin by the competitive inhibitions. So in the competitive inhibition, we have enzyme A, and red color is in substrate, and this yellow color something molecule is inhibitor. So actually the shape of the inhibitor is as similar as the substrate molecule, and they are morphologically, they both are similar, but chemically they are different. And this outsider inhibitor, it starts competing with the substrate molecule, and they both try to bind with the enzyme's active site by the process of diffusion. And the binding also occur because of this shape, and their shapes are similar, but it doesn't mean that their chemical nature is not going to affect this binding. They are of course going to affect it somehow. If the substrate binds with the enzyme's active site, it will be a tight binding. But if suppose the inhibitor tries to bind with the enzyme's active site, it will be a loose binding because they are chemically different. No chemical reaction going to be occur at that point. And that is the reason why it's a reversible process. And it has also been asked in the NEET exam that whenever an enzyme tries to bind with the inhibitor to form the enzyme inhibitor complex, inhibitor can lose its binding because of its loose binding error. Now the question is, how can we overcome this? Is there any way? Then yes. See, if we have 100 substrate molecules and 10 inhibitors out of 100 substrate molecules, then the chances of inhibition is 1 by 100. But what if we increase the number of substrate from 100 to 1000? Then the chances of binding will automatically increase rapidly by 1 by 1000, that's the only way by which you can overcome this competitive inhibition. And that also plays an impact on the number of Vmax and Km, but the Vmax will be unchanged because we didn't interacted in the number of enzymes in by increasing or decreasing it, so it will be unchanged. No change will be there in the Vmax, but Km will be increased. But the Km will be increased because we increase the substrate concentration for the same velocity of enzymes. And we also have an example when the succinate converts into the fumarate by the help of succinate dehydrogenase enzyme, where malonate as an inhibitor tries to compete with the succinate, which is an substrate, for the succinate dehydrogenase binding to form the product fumarate. And such competitive inhibitors are often used in the control of bacterial pathogens. Let's see what our NCERT says about this. So When the inhibitor closely resembles the substrate in its molecular structure and inhibits the activity of the enzyme, it is known as what? Competitive inhibitor. And due to its close structural similarity with the substrate to the inhibitor start competing with the substrate for the substrate binding site of the enzyme. Consequently, the substrate can't bind. As a result, the enzyme's active site action they decline. Now we have non-competitive inhibitors. So in this case, inhibitors doesn't binds with the enzyme's active site, but it tries to bind with some other allosteric sites of the enzyme, which cause in a change of enzyme shape and can reduce its activity and even can alter its structure, by which enzyme can lose its functionality regardless of substrate, because in this case inhibitor is not in the competition with substrate. As the name suggests, it's a non-competitive inhibitor, so substrate is going to bind with the active site as always. Now we will see what are the possible ways to overcome this issue. So we can remove the inhibitor by any of the chemical treatment, or and in this case inhibitors are going to affect Vmax. See, as the inhibitors affected the enzyme's functionality, so the number of enzyme will going to decrease, so the Vmax will also be decreased and the Km will be unchanged, because we find Km for the half of the Vmax. And suppose if we have 100 enzymes, then inhibitor going to destroy 5 or 10 out of them, but still we have 90 left. That may can affect the value of Vmax but cannot affect the Km. Now we have this example when chemical cyanide as an inhibitor binds with this cytochrome oxidase enzyme, which is also called complex 4 in the etc. Then you must heard about this chemical called cyanide before, but you may not know how it can cause death within a second. So let's see. Once cyanide enters the body, it reaches to the cell, especially mitochondria, and when cyanide binds with the enzyme called cytochrome oxidase, it actually starts its work from here. It blocks all the paths of oxygen uses in the cell, and without oxygen, cell struggles to produce ATP and leads to the failure of many essential processes and many metabolic process and can cause instant death. The effects of cyanide are not limited to a single organ or tissue. It's a deadly poison, can cause rapid death. There is one more example. If you remember the series Family Man, in the season where Srikant was on a mission to prevent the nerve gas attack in Delhi, nerve gas also shows this non-competitive behavior where it binds with the acetylcholine enzyme in the nerves and can affect the whole neurological system and can cause death within seconds, because it's one of the most deadly weapon. And I forget to say, this process is also reversible like the previous one. Now we have uncompetitive inhibition, which is completely different from the non-competitive inhibition. In the non-competitive one, there was some situation or activity that doesn't allow inhibitors to involve into the competition with substrate. But in this uncompetitive inhibition, the inhibitors are not capable of competing with the substrate. In this mechanism, inhibitors only binds with the enzyme substrate complex to prevent the reaction from completing, to form inactive ESI, enzyme substrate inhibitor complex. And why I said inactive? Because they can't make products. Now we can overcome this by removing the inhibitor physically or chemically, but it becomes very difficult to remove the inhibitor, because since the inhibitor binds only after the substrate is already bounded to the enzyme's active site. Also, there are few other ways by which you can overcome this. Okay, now we'll see how uncompetitive inhibition can impact on Vmax and Km, so they both are decreased. See, as we already know that ESI complex can't make products because they are inactive. So, after having a lot of substrates and enzyme, the enzyme system cannot reach its normal maximum speed called Vmax. That is the reason why Vmax decreases. And Km will also decrease because the system reaches half of the Vmax speed at a low substrate concentration. That is the reason why Km decreases. Then we have this example when the substrate phosphate ester binds with the enzyme alkaline phosphatase plus inhibitor phenyl and aline, they will form ESI, inactive enzyme substrate inhibitor complex. And also this process is reversible, but only after the ES complex forms. Then we have feedback inhibition, where the product of reaction tries to stop its own conversion. What happens here is when the final end product forms in the metabolic pathway, it binds to the earlier enzyme in the pathway and prevent the enzyme from working and slows down to stop the entire pathway. It helps organism to maintain their internal balance by monitoring or adjusting their internal environment within biological system by regulating the rate of metabolic pathways. So this is the first step of glycolysis, where glucose bonds with the ATP by the help of hexokinase enzyme. It forms glucose 6-phosphate and ADP, where the final product binds with the enzyme and inhibits its production. And also there are two type feedback inhibitions called negative or positive feedback inhibition, but this is not in your NCERT for now, so we are skipping it. And also these feedback inhibitions are reversible like the others, just to stop the overproduction of the product formation. Now we have classification and nomenclature of enzyme. So thousands of enzymes have been discovered till now. Out of these enzymes name follow a structural system based on which reaction they catalyze. There are mainly two different types of system. First is common name, which you are already familiar of, our common traditional system, which basically ends with A's, and their names are based on the substrate and the types of reaction. That is the reason why their names are easy to remember. For the example, we can take enzymes like lactase, which breaks down lactose, proteases, which break down proteins, and lipase, which break down lipids, and DNA polymerase, which synthesize DNA strands. Then we have systematic names given by IUBMB, which is International Union of Biochemistry and Molecular Biology, which gives a clear description of the reaction. And most of the enzymes have been classified into different groups based on the type of reactions they catalyze. And basically these enzymes are divided into 6 classes, each with the 4 to 13 subclasses, and named accordingly by a 4-digit EC number. EC number means enzyme commission number, we'll see. And also it follows a systematic name structure, where substrate lies first, bonds with the reaction type, and ends with the A's. As you can see in the example, we have substrate as a lactase, reaction type is dehydrogenase, and ends with A's, dehydrogenase, and which removes hydrogen from the lactase. We'll see. Okay, so these are the systematic naming of our 6 classes called orthollele. You can remember this mnemonic orthollele. See, orthollele stands for O stands for oxidoreductase, T stands for transferase, and H stands for hydrolase, L stands for lysase, I stands for isomerase, and L stands for ligases. And learn these names consecutively according to their enzyme commission number. I mean, oxidoreductase stands for EC1 and transferase stands for EC2. So you can't exchange it with EC2 and EC1 because these enzyme classes, the names are fixed with their specific EC number. Let's see these 6 enzyme classes in detail and what are the reactions they catalyze. Now we have oxidoreductase, also called as dehydrogenase, which catalyze redox reaction. Redox reaction means the reaction in which oxidation and reduction both occur simultaneously by the help of electron transferred between the molecules. As you can see, we have this example on the screen. We have two substrates called S and S-. On the left side of the reaction, we have S as a reduced and S- is oxidized. After the reaction, when electron transfer occur from S to S-, S is oxidized and S- is reduced by the oxidoreduction reaction, where the electrons are transferred from one molecule, the reductant, to the another molecule called oxidant. As you can see this example, when lactate is converted into pyruvate by the help of lactate dehydrogenase enzyme, where the transfer of electrons take place from NAD to NADH. Then we have transferase, where enzyme catalyze a transfer of functional group from one molecule to another molecule other than hydrogen, because transferase do not deal with the transfer of electrons or hydrogen atoms like oxidoreductase does. Then there will be no difference, can't you see? And instead transferase deals with, they are moving the entire functional group, not just an atom. Now again we have this pair of substrate called S and S dash. As you can see in this example, when a compound is comprising of S and G, and when they react with the compound C, then you can see there is a transfer of G group from Sg to the compound S dash. Let me take you through an example from the mineral nutrition chapter. This amino transferase enzyme catalyze the transfer of an amino group NH2 from one molecule to the another molecule to form a new amino acid and a new keto acid, where they are usually amino and keto acids. Look at the example given above the screen. When NH2 is transferred from alanine to alpha ketoglutarate by the help of aminotransferase enzyme, alanine becomes pyruvate, which is a keto acid, and alpha ketoglutarate becomes glutamate, which is an amino acid. Then we have hydrolases, where enzyme catalyze the hydrolysis of various bonds using water. Bonds like ether bond, ester bond, CC bond, PN bonds, and many other bonds written on the screen you can see. As you can see, we have this general term where we have the compound A or B in the presence of H2O. It hydrolyzes the reaction by breaking down the compounds into two or more compounds. Now we have this example of trypsin, which is a protease or proteinaceous, also called proteolytic enzyme, which breaks protein in the presence of water into peptide bonds during digestion. Then we have liage, where the enzyme catalyzes the addition or removal of the group from the substrate to form double bonds or break the bonds without any use of water or redox reaction. Suppose if we have this compound X or Y, if we remove this, then we'll get double bond between these carbons. And always remember that X and Y should not be water. Then we have this example, pyruvate dehydrogenase enzyme, which removes CO2 from pyruvate to form acetaldehyde without using any water. Now we have isomerase, where they catalyze the intermolecular rearrangements, also called isomerization, by converting a molecule from one isomer to the another. And it includes all the enzymes catalyzing interconversions of optical, geometric, or positional isomers. Anyways, we have this compound A and B, which converts into B and A, where we have same atoms but the different arrangements. You can also take this example of phosphoglucoisomerase enzyme, which convert glucose 6-phosphate G6P into fructose 6-phosphate, which is F6P, in the process of glycolysis. Then we have ligases, last but not the least. Those are the enzymes which catalyze the linking together of two compounds by using ATP or similar energy sources. And surprisingly, this is the only class of enzyme ligases which act as the joining catalyst. It links the bond of carbon with oxygen and carbon sulfur, carbon nitrogen, phosphorus oxygen, and many more. As you can see in this, AB molecules use ATP for the synthesis of AB compound and turns into ADP. Now we all know enzymes are chemically composed of proteins, but in some cases these proteinaceous enzymes are not enough to catalyze functions. In that cases, some non-proteinaceous constituents, which is called cofactors, are bound to the enzyme to make enzyme catalytically active. So if we talk about the chemical nature of this complete hollow enzyme structure, then first we have apoenzyme, which is proteinaceous in nature, and as it has protein, it is organic in nature, and we already know this from the starting of this lecture. The only new thing we have here is cofactor, and these cofactors are they are of chemically two types. They are organic and inorganic. We have subdivisions of cofactor. We have prosthetic group and coenzymes as organic cofactor, and metal ions as inorganic cofactor. We will see them in detail, but before, let's observe this diagram once. So this is our regular protein enzyme, which is now called as apoenzyme because of its proteinaceous nature, which is in the blue color. Now, other than this apoenzyme, all the labeled diagrams you can see in this figure are non-proteinaceous. As you can see, this is our inorganic cofactor in the yellow color, and this is the organic cofactor, which is in orange color. So this inorganic cofactor is they could be metal ions, and these organic cofactors, they are coenzyme as written, but they can be prosthetic group also, as I already said, and this is our regular active site. Now we have a detailed breakdown of organic compounds. As the name suggests, they are carbon containing molecules which are usually derived from the vitamins. So for the very first, we have prosthetic group. They are the tightly bound cofactors and is permanently attached to the enzyme surface through the covalent bonds and stay bounded through the multiple catalytic cycles throughout the reaction. As they are tightly bounded to the structure of enzyme, if you ever try to remove these prosthetic groups from the enzyme structure will be completely denatured, and we get them from the sources like vitamin B7 and some iron containing elements. Then we have this example of peroxidase and catalase. These both are the enzymes that catalyze the breakdown of hydrogen peroxidase, which is H2O2, which is a very reactive and harmful byproduct of cellular metabolism, you will see in that chapter, and breaks down into water and oxygen, where heme, iron containing heme, act as the prosthetic group in that reaction. And this reaction helps to protect the cell from the oxidative damage caused by hydrogen peroxides. Don't be confused with the examples. I have not taken it from any other sites. These are only from your NCERT. Then we have another organic cofactor, which is coenzyme. Their names are quite similar, so it becomes easy to remember. They are loosely bound cofactors that is temporarily attached to the enzyme's active site. They bind transiently during the reaction only. It means they enter the active site, participate in the reaction by donating, accepting electron, atom, or any functional group or whatever, and then they leave as soon as the reaction is over. They just bind for few moments only. They are generated from vitamins and vitamin derivatives like vitamin B3, B2. Then we have these examples of NAD, which is nicotinamide adenine dinucleotide, and NADP, which is nicotinamide adenine dinucleotide phosphate. So both of these are the essential coenzymes that contain the vitamin niacin, which is also called vitamin B3, as a key structural component. Then we have this inorganic cofactor, which is metal ion. See, a number of enzymes are there which require metal ions for their activity, which form coordination bonds with the side chains of the active site, and at the same time form one or more coordination bonds with the substrate also. Sorry, we have few other enzymes which are other than from this chapter, like Rubisco DNA polymerase, which required Mg+2 as a metal ion, and catalase, which is heme, which require iron, and nitrate reductase, which requires molybdenum. And then we have carbonic anhydrase, which requires Zn+2. We have this example where peptide bond is broken by the hydrolysis of a water molecule with the help of cofactor zinc for the proteolytic enzyme called carboxypeptidase to form simplest amino acids. You already know this. It plays a key role in the digestion of proteins, especially in the pancreas and the digestive tracts. And yes, some enzymes need both coenzymes and metal ions to be active, so both are equally important. And yeah, one more important thing I forget to tell you, that once you remove the cofactor from the active site of the enzyme can lost all its activity and even can be denatured. This simply says cofactors play a crucial role in the catalytic activity of the enzyme. You will see many of these questions related to cofactors in your board exams and other competitive exams. So all the best, everyone. Okay, so thank you everyone for being till the end, and you can download the mind map of this chapter from the description, and Hope you all have a good day. If you're watching it at the night, then have a good night. See you soon in the next lecture.